Hey everyone, this is your Ornithine cycle for urea video. This cycle has to do with proteins. Obviously, we're in the protein unit, but specifically when we are breaking down proteins, so when we're doing proteolysis. So remember that proteolysis is just catabolism or breaking down of proteins. And we'll be doing this when we're in the starvation, not fed state. So unfed state. And remember, this doesn't have to be an extreme level of starvation, although of course you do more, the more extreme your level of starvation is. But this could just be general, fasting as you're sleeping or haven't eaten for a while. In this stage, you start breaking down muscle proteins. Remember, the muscles are giving off, various amino acids. We mentioned that before about how we had the various, amino acids being given off by the muscle cells as they're breaking down their proteins. Now, those proteins are being used for energy. They're being used, if they're ketogenic, they're being used to create keto bodies. If they're glucogenic, they're being used to create glucose through gluconeogenesis, and also acetyl-CoA to run through the Krebs cycle. But there is a byproduct of that, which is ammonia, so NH3, and this is toxic. So this is not something that you want to have accumulating in the body, so you have to be able to do something with that ammonia. So what you're going to do is you're going to convert a lot of that ammonia into urea, and urea is excreted by your kidneys. Now, any ammonia that is not excreted by the kidneys, that is not turned into urea to be excreted by the kidneys, some of that ammonia can also be excreted by the kidneys itself, but it can also be used in producing glutamine, and we know that glutamine is related to glutamate, and then we can go into all sorts of different pathways from there. So we can also kind of recycle some of that NH3, but we're going to not want to have a whole lot of that. We're going to want to turn it into urea and get rid of it. Now, for the ornithine cycle for urea, this is also called the Krebs-Hanselite cycle. So notice Krebs. Krebs was, busy working on metabolism, and here he was collaborating with Hanselite to describe the way that you create urea out of ammonia. And what they discovered was that there are two ways to enter this cycle. It's a cycle that really, it's a little circle. We have an entrance here. This would be, the first entrance, I guess. It doesn't really, it's just arbitrary to say that's the first one, but this is the first one we're going to talk about. And this would be the trans-deamination entry, or the trans-deamination route. And then there's another entry point over here that is just the plain old trans-amination route. So both of these are ways that we can enter into this ornithine cycle to turn our potentially toxic byproducts into urea. And just get into this ornithine cycle. Where we'll have urea as a product. We're also going to have fumarate coming off of here. Keep your eye on that. That's going to pop up again in the last couple of slides here, because there is a use for this where we can kind of recycle and turn this back into energy production, rather than just have this be a cycle all about waste. We can actually, tie this back into energy production. So here's our cycle, and we're looking specifically at the trans-deamination route. Now, this word tells you exactly what's going to happen. First, we're going to transaminate, so we're going to take an amino group off of something and put it onto something else. Then we're going to deaminate. So we're going to just remove the amino group. So we're going to have two steps for this. Now, the first parts of this is happening in the mitochondria. This part here is the ornithine cycle for urea to create urea, and this is in the mitochondria. But we have to prep it first, and that will be happening out in the cytoplasm. So use a different color for the cytoplasm stuff. So we're going to see a molecule that we have seen a lot of times before, and it's alpha-ketoglutarate. Oops, alpha-ketoglutarate. And we're going to add an amino group to it. So transamination occurs. This is the transamination part. And it's catalyzed by aminotransferase. So we've seen that enzyme before. And what our product is going to be is glutamate. That's another thing we see before. Okay, so we can see again how tied together all of these things are, because where have we seen alpha-ketoglutarate? We've seen it in the malate aspartate cycle. We've seen it in the Krebs cycle. We've seen it all over the place, right? We've seen glutamate with the production of other proteins, with transamination in the liver and things like that. We've seen, and coming out of skeletal muscle, we see glutamate in a lot of different places. So here we are just kind of tying everything together and linking it to like, okay, we have waste products now, let's get rid of our waste products. Okay, so what happens now is the glutamate itself is going to be able to be carried into the mitochondria, and it's going to go, I'll put it over here so I have room. So in the mitochondria, and now we're going to have our glutamate get deaminated. So we have glutamate minus our NH3. And this gives us, or I shouldn't say equals, I should say yields. It gives us alpha-ketoglutarate again. Plus ammonia. Okay, and you might go, okay, that's what we started with. Why did we do any of that with the glutamate in the first place? And the reason is just to get that ammonia into the mitochondria for the ornithine cycle. We can't just transport the ammonia straight into the mitochondria. Glutamate is essentially the thing that's going to carry that amino acid into the mitochondria. Just like we saw in our first unit, we saw things being used to transfer hydrogens into the mitochondria, or NADH, to create NADH again within the mitochondria. So we're just taking a hydrogen and moving it across by adding it to another molecule that can cross the mitochondrial membrane. This is the same thing. We're just adding this ammonia to alpha-ketoglutarate so we can make glutamate that can cross the mitochondrial membrane, and then we take it back apart again. And that leaves us with alpha-ketoglutarate. We can use that in the Krebs cycle. Or, as the reverse of this in the malate aspartate cycle, we could do that, reaminate it, deaminate it, back and forth. But now we've, whatever we've done, we've left our NH3, our ammonia, in the mitochondria. Now, this reaction is catalyzed by glutamate dehydrogenase. So let's just put that down. Okay, so that's what takes the NH3 off of the glutamate and gives us our ammonia. Now we can leave alpha-ketoglutarate alone. It's off to do its own thing. We are worried now only about the ammonia. And what we're going to do is we're going to use the ammonia to create another molecule, this one, carbamoyl phosphate, that can go into the ornithine cycle. So ammonia creates carbamoyl phosphate, And this is catalyzed by carbamoyl phosphate synthase, which is an enzyme. So carbamoyl phosphate synthase is our enzyme, but this is our product that we're looking for. There it is right there. Entering into the ornithine cycle, there's ornithine. That's the final step of the cycle. We add the carbamoyl phosphate and we get citrulline. Then we go on through all the way around. We give off urea. We also give off fumarate. Remember, we want to remember fumarate for a little bit later. But our main point is urea is going to go to the kidneys. The kidneys to be excreted. Okay, so now we have done the trans-deamination route. We have taken our ammonia, we've transferred it to glutamate, then we put the glutamate in the mitochondria, we deaminated it, and we turned that ammonia into carbamoyl phosphate to go into the ornithine cycle. So that's one way of entering the ornithine cycle. The other way of entering the ornithine cycle is right around here, where we're going to just have the transamination route, so no deamination involved here. And this is just going to involve our first step all over again. So here out in the cytoplasm, we have again alpha ketoglutarate. Plus NH3, so we're transaminating here, and this is again aminotransferase. Okay, and we get glutamate. Okay, now the glutamate again carried into the mitochondria. Just like it was before, but instead of being deaminated, we're going to transaminate again. So we have our glutamate, and we're going to convert it into aspartate. And the acceptor of that amino acid is oxaloacetate. So oxaloacetate, we are going to transaminate, so we take the amino group off of glutamate and we add it to our oxaloacetate, which gives us aspartate. And we've seen this reaction before, again, in that malate aspartate cycle. We've seen, the transamination between glutamate, aspartate, oxaloacetate, alpha ketoglutarate, all of those things just bouncing amino groups off of each other, or trading amino groups from one to the other. Now the enzyme for this is aspartate aminotransferase. And this is going to go into the cycle. So here, right there, we have the aspartate react with citrulline, which was, what we created from, carbamoyl phosphate when we entered from the trans-deamination route. But now we already have the citrulline and we are entering from the transamination route instead. And we just react the aspartate with the citrulline and we enter the cycle just like always. We get urea for the kidneys to excrete. We get fumarate, which is going to tie us back to energy creation. Same cycle, different place, right? Okay. So, now that we know the ornithine cycle for urea, it's really quite a straightforward little cycle. But we also need to think about how this ties back to, say, skeletal muscle and energy production or energy storage. So one thing that we need to look at is the arginine. So the arginine is part of this cycle. It's, a step in the cycle, but some of that arginine, instead of just continuing on in the ornithine cycle, it can go into a creatine phosphate pathway. Remember that creatine phosphate is the main source of stored energy, or the main form of stored energy in skeletal muscle. So it's the main form of stored energy in skeletal muscle. So you'll have some ATP in skeletal muscle, but more creatine phosphate. So really it works in the same way that ATP does. You're just putting a phosphate group onto a molecule to store the energy, and then you take the phosphate group off in order to release that energy and use it. So we just go through a process here. We don't need to go through every enzyme of this process. The whole point is just to see how we tie the ornithine cycle, so the process tied up with the breakdown of actual muscle mass, muscle proteins being broken down and giving off ammonia, with tying it back into, okay, now we're entering into like the fed state and we're going to store energy instead, or if we're remaining in the starved state, okay, we're going to send some of that energy back to the skeletal muscles so they can store the energy and use that energy even though we're in the starving state. So you still need energy even if you're, starving. You still need to do stuff, right? So there's our arginine. Note the little amino acid there. We got some little amino groups here. And we're going to enter into this creatine phosphate cycle or pathway. It's not a cycle, it's not a circle, but the creatine phosphate pathway. And this is happening in cells in the kidney, right? There's ornithine given off by this, so we can go right back into our ornithine cycle there. So just like, we did right here. And here, from there, that ornithine can go to the liver. So here in the liver, we can use energy. So ATP being converted into ADP. We're using energy here to create creatine phosphate. So that PI, the energy from the ATP, was taken off and it was added to creatine, to make creatine phosphate. This is the stored form of energy. There it is, right there. And when we want to get that energy out, so when the skeletal muscle contracts, we break this off of the creatine phosphate. So there it goes, off. We have energy. Use it to have a muscle contraction, and that leaves us with creatinine. Creatinine is, can have another phosphate added onto it. It can be excreted by the kidneys again, so go out into the blood and be excreted by the kidneys. The creatinine can be reused to make more creatine phosphate. So multiple things you can do with the creatine. But the main point here is that we essentially put an energy accepting molecule of creatine into the skeletal muscle and allowed energy to be produced, or energy to be stored and then used by the skeletal muscle. And that was all coming, sort of a return of energy from the ornithine cycle from arginine. Okay, now also remember our fumarate. So fumarate was part of our cycle, something given off by the cycle. So not a part of the cycle itself, but given off by the cycle. But we can use the fumarate as sort of an alternative towards working the Krebs cycle. So remember in the Krebs cycle itself that we have the molecule oxaloacetate. So oxaloacetate is kind of at the end of the Krebs cycle, and then we react our acetyl-CoA with it to get citrate, and then we're, going through the Krebs cycle. But if you are doing the Krebs cycle a lot, maybe you have run out of oxaloacetate. Maybe you don't have enough of a supply of oxaloacetate to run the Krebs cycle as much as you're trying to with the acetyl-CoA. So in that case, this would become a rate-limiting substance here. So if you don't have enough oxaloacetate, then you can't do the Krebs cycle even if you have plenty of acetyl-CoA. So when you're exercising, when your muscles are being used, they're using energy at a rapid clip. They're breaking off, creatine or phosphates from creatine phosphate. They've used up their store of ATP within like 2 seconds. So they end up needing to create energy quite quickly. And they can then run low on oxaloacetate and then start getting into that state where you're unable to do aerobic respiration, not just because you've run out of oxygen, although that can also be a cause, but because you've run out of oxaloacetate, and that would get you stuck in glycolysis and with a risk of lactic acidosis and all that stuff. And also just less energy. So none of that is good. So instead, we can use fumarate and convert it so that we can run the Krebs cycle again. So what we're going to do is use this purine nucleotide cycle. And we take the fumarate, we convert it to malate. So there's malate again. We've seen malate a million times too, right? And for malate, we can easily get to oxaloacetate through this reaction. Okay, so we just need this enzyme here. This is a reversible reaction. Once we have the malate, we're going to run it this way, and it's just going to, utilizing NAD plus and NADH, having its hydrogen being removed as part of this reaction, but we get oxaloacetate in return, and then we can run our Krebs cycle again. Okay, so that's tied from the ornithine cycle. We've gone from, breaking down ammonia, essentially, or converting ammonia to urea, but this can go off to help us run the Krebs cycle in our skeletal muscles that are in need of it. Okay, so here on this slide, this is just going into a little bit more detail about that purine nucleotide cycle, and we're just looking at the fumarate here. The adenylyl succinate, that comes from that ornithine cycle. Okay, so that is, or I'm sorry, not the adenylyl cycle. The fumarate is coming from the ornithine cycle right there. Not the adenylyl succinate, that's just another molecule, but the fumarate's coming from the ornithine cycle. And here we have aspartate, right? That's aspartate that's also tied up in the ornithine cycle. I should just leave that page out. Okay, so aspartate could be entering into the ornithine cycle. Here we're giving off energy, right? So energy is being used there. AMP, if you think of cyclic AMP or CAMP, right, this is a stimulatory kind of enzyme. This is something that happens as you have ADP being broken down further. If you were to add the cyclic part, this would become a stimulating kind of enzyme, but without it, it's just going to be able to become part of this purine nucleotide cycle. So we can either add a phosphate to create ATP for stored energy, or we can remove phosphate for this particular molecule. Now, these have amino acids on them, so we can deaminate them. There's ammonia. So the ammonia can go to the ornithine cycle. And we're just going through this whole entire system here to get even more fumarate in addition to the fumarate that has come from our ornithine cycle. Okay, so essentially, that was a very long, confusing explanation of really what is the important part in a nutshell. You take water and aspartate, so over here, we add in water. We use GTP, we end up with GDP. We've used some energy there, so here's energy used. We end up with ammonia. That goes to the ornithine cycle, GDP, and then we have the energy being used, and we get fumarate. And from the fumarate, we can then get malate, which can then go into the oxaloacetate and become part of the Krebs cycle. Okay, so that is the main gist. I know I wasn't super clear about that, so I'm just going to write the main gist here, that the fumarate in the purine nucleotide cycle is used to replenish oxaloacetate via malate, and then turning into oxaloacetate. In the process of this, so in the whole process of doing all of this, more ammonia is created for the ornithine cycle. Energy is used, but more energy will be created, so that's okay. Okay, so we're just tying everything together here. We got fumarate from the ornithine cycle. Now we have ammonia for the ornithine cycle. This is a waste product. This is a good thing. This will help us create energy. So that's the take-home message from that. Okay, so that does it for the ornithine cycle for urea.